In this video, we describe a method of fitting spheres to isosurfaces of electron density. We consider this problem because applications like protein folding and docking prefer models where atoms are perfect spheres, while tools that determine structure of proteins generally produce electron density maps. There are two sources of electron density maps: quantum calculations that produce high quality maps for small proteins, and X-ray crystallography that can handle large molecules but produces incomplete maps. For example, some hydrogen atoms may be missing in a X-ray crystallography map. The hydrogens are then added using simple geometric rules that depend on geometric parameters like distance from the attached atom. We determine the values of these parameters from the high quality quantum maps and then use them to verify the accuracy of the values in molecular biology toolboxes like REDUCE and SHELEX. Electron density is a real valued function over 3D space. The structure shown here is an isosurface of electron density. As the associated electron density value is increased, the isosurface shrinks. However, it still roughly resembles the surface of a collection of spheres. We find spherical patches on such surfaces and associate these patches with atoms of the protein. Here we have colored the isosurface by curvature. Red means higher curvature while blue means lower. We use a non-standard measure of curvature which is defined in the abstract. The key observation behind our sphere fitting method is that spherical patches are contiguous regions of high curvature bounded by regions of low or negative curvature. Based on this observation, we could do a simple thresholding to discard the points of low curvature, and hopefully this segments the isosurface into individual spherical patches. However, some boundaries are less pronounced than others, and hence the same threshold may not work well everywhere. Instead, we use a join tree to keep track of the merging of patches as we reduce the curvature threshold. The glowing region here joins two patches into one larger patch. The join tree records all such events in a tree data structure. Each node of the tree corresponds to a patch on the isosurface. The node 398 here corresponds to the patch shown in red. The root node corresponds to the whole isosurface. The patch corresponding to a parent node is a superset of its children patches. From this tree, we can manually or automatically choose spherical patches. To each chosen spherical patch, we fit a sphere using a simple linear program. We now use the results from our sphere fitting method to compare the hydrogen placements in the toolboxes REDUCE and SHELEX. REDUCE and SHELEX place hydrogen spheres based on expert knowledge. The edge skeleton shown here is the structure determined by REDUCE. The vertices correspond to centers of atoms and edges correspond to bonds. Our method identifies a set of dots for each of the 4 hydrogens in the structure. The ball shown here is the center computed by our method, while the red triangle is the center determined by SHELEX. REDUCE places the hydrogen at the free end of the bond. We find that SHELEX consistently matches our calculations better than REDUCE does. Here is an example of our results for a larger molecule. Thanks for watching.